Hello. Thanks for joining us today for our fourth webinar in a series on bioavailability-enhancing technologies. My name is David Bodak. I'm Vice President here at Bend Research. And I'm going to introduce a really exciting topic today on nanotechnology for bioavailability enhancement. As I said, this is the fourth in a series, and we've gotten a lot of client feedback that it would be great to educate everyone on how we and others in the industry think about selecting from all of the different technology options that are enabling in advancing low solubility compounds and compounds that require different enabling aspects to get the required absorption for efficacy. So today we're joined by our speaker and my colleague, Mike Morgan. As introduction, Mike has been at Bend Research since 2003. He brings more than 15 years of experience in the development and evaluation of novel polymeric materials and of pharmaceutical formulations for oral and non-oral delivery, particularly for low solubility molecules. He received his doctorate in physical chemistry from the University of California at Berkeley and is co-inventor on 13 patents and patent applications and has authored over 20 papers. So with that, I'd like to get right into the outline for this webinar presentation. Initially, I'm going to do a brief introduction to the Capsugel dosage form solutions and Bend Research and how that division is set up to be a solutions provider in the pharmaceutical industry. I'll get into a brief, and then I'll turn it over to Mike, and he'll give us a brief overview of the general nanotechnologies for solubility and dissolution rate enhancement, and then dive into some fundamentals around each of the technologies, touching on nanocrystalline and amorphous embodiments of these nano systems. And then finally, Mike will wrap up with some really interesting case studies that highlight the enabling pieces of each of the technologies. So Bend Research is now part of Capsugel's Dosage Form Solutions, which is a business unit headquartered in Morristown, New Jersey. And the two companies combined have brought a wide range of technology and solutions to be able to be brought to bear to the pharmaceutical industry. Briefly, Bend Research is a leading scientific R&D and manufacturing company, as I said, that's now part of Capsugel, and it really is, we think of ourselves as a problem-solving organization to help our clients get their products to the market and solving some of the harder problems in the industry with our different technology platforms. And one of the key aspects for this new division, Dosage Form Solutions from Capsugel, is this incredibly large toolkit of technology platforms. And the way we see the world is in these different areas, bioavailability enhancement, which we're going to dive deep in on today, but we also have technologies in the modified and targeted release, as well as some other specialized healthcare applications. In the bioavailability enhancing area, the main technologies that we utilize are in the amorphous, nano microcrystalline, and lipid and liquid-based technologies. And with that wide range of technology offerings, we're able to really mate up the right technology with the problem statement that the client has. And we're not going to push a given technology because that's what we have the most experience or the most to gain in, and I think that gives our clients a lot of options. And in fact, with these, we use these tools, and in front of them have guidance maps, and use that to take a client's target product profile and product concept in conjunction with the API properties and the pharmacokinetic requirements, and we use that to, in conjunction with our technology selection guidance maps, to select the appropriate technology or technologies, and then take that with our client to say what is the enabling formulation and technology that we need going forward. And then with these technologies, we have the ability to utilize these platforms from feasibility and formulation identification through development, scale up, and ultimately commercialization. So with that, I'd like to turn it over to Mike and get started.
All right, thank you, David, and thanks to our audience for joining us today for the webinar. As David mentioned, the topic of today's webinar is around nanotechnology for oral bioavailability enhancement. And of course, nanotechnology has received a lot of interest lately, both in the pharmaceutical industry as well as other industries. And in the pharma area, a wide range of nanostructures have been investigated for drug formulation and delivery. A number of classes of those structures are shown schematically here. A number of these have been used preclinically, clinically, and commercially for a variety of oral and non-oral delivery applications. Today I was going to focus on two of these that have shown utility for increasing oral bioavailability, namely nano-sized drug crystals and then amorphous drug-polymer nanoparticle dispersions. So some of the other nanostructures, such as those consisting of lipids, will be covered in a future presentation. The drug crystals, shown here schematically, can be made by either treating larger crystals or by growing crystals from solution and arresting the growth before they get large. And so these are commonly known as the top-down and bottom-up approaches, respectively. And there are several ways to accomplish each of these, and I'll talk a little bit about both. And likewise, there are different processes for making the drug polymer nanoparticle dispersions, and I'll mention a couple of those in the presentation as well. So when thinking about oral absorption of drugs, it can be useful to categorize the delivery applications, and one tool for doing so is the so-called Developability classification system introduced by Dressman and co-workers. Many of you are familiar with this. The DCS is a modification of the original biopharmaceutics classification system, and there are four classifications in which Class 2 is low solubility, high permeability compounds, and in the DCS class system, Class 2 is further divided into 2A, which contains compounds whose solubility and permeability relative to dose renders them dissolution rate limited, and Class 2B, which have lower solubility and or permeability relative to dose. So the distinction between Class 2A and 2B can be important because some formulation approaches provide increased dissolution rate, and some provide solubilization, i.e. higher dissolved drug concentrations, and of course some provide enhancement in both dissolution rate and dissolved drug levels. And then the Class 4 compounds have low solubility and permeability relative to dose, and everything there is solubility limited. So you can see from the percentages shown here on the slide that a large fraction of compounds in development are in either Class 2 or 4, so having approaches for delivering them is important. So I'll return a little bit later to the DCS map in a later slide. So the DCS categorizes drugs by solubility and permeability. We use various other maps to guide formulation selection as well, and one of those is shown here. And what is shown is a melting point on the y-axis versus the log of the octanol water partition coefficient. And this map can give guidance on choosing technologies to achieve both performance and physical stability of the formulation. So note that drug compounds can have low aqueous solubility either because they have strong crystalline forces, as evidenced by a high melting point, or because they have high lipophilicity. Generally speaking, the nanocrystalline formulations that we'll talk about are primarily used for high melting point compounds because these compounds have a strong tendency to crystallize, and have low solubility not just in water but in most solvents, and therefore are difficult to stabilize as solid or liquid solutions or other amorphous forms. Drug polymer dispersions using high TG polymers can be used for the same low log P high melting point compounds, but the loading often has to be restricted to maintain physical stability of the dispersion. So these types of dispersions tend to be more readily applicable to compounds having a little bit lower melting point or higher log P because they're amenable to making relatively stable dispersions with high drug loading. For compounds that are extremely lipophilic, down here in the right lower corner of the map, drug polymer dispersions can be useful, but often these compounds can have a low tendency to crystallize, and the amorphous state can be maintained by low TG polymers, lipids, or in extreme cases, just maybe a drug that's in oil form stabilized in a nano-sized droplet phase by a surfactant. So it's important to recognize that despite what I just said, this is a simple 2D map of a multidimensional problem, but it can provide a rational starting point for choice of formulation technologies. Some of you have probably tuned into previous webinars and saw David Vodak's initial map here on technology guidance, and so I just wanted to show this again to relate the nanotechnologies we'll be describing today to some of the other approaches that were introduced previously. And so the nanocrystals are called out specifically on this map, and then the other two, the amorphous drug polymer nanoparticles, as well as the bottom-up nanocrystals that we'll talk about, more or less lie mostly in the area labeled amorphous and overlap with each other on this map where it says amorphous. And like the previous map, this one is not meant to suggest that there are sharp delineations where technologies are to be applied, but gives some guidance on a formulation starting point. So the various maps are complementary ways of sort of categorizing delivery challenges. So as I mentioned, nano-sized drug crystals can be made by the attrition, that is top-down approach, or by precipitation from solution in a bottom-up approach. For the top-down approach, there are several processes that have been used. Probably the most common one is a wet milling approach, often referred to either as wet ball milling, bead milling, or pearl milling, in which a grinding media is added to crystalline drug suspended in a non-solvent, and then energy is added to attrit the crystals. This is the approach used in the well-known Elan nanocrystal process, for instance. Now, instead of using grinding media, one can also use the shear and cavitation present in homogenization using either a piston gap instrument or a microfluidics process, for example. And the Dissocubes and nano-edge approaches are examples of these respective processes. The nano-edge is actually a hybrid of the top-down and bottom-up approaches, as it involves precipitation of crystals followed by an attrition process in a homogenizer. The bottom-up approach typically involves nucleating, growing, and then arresting the growth of small crystals from a supersaturated solution. The solution is often a liquid, but turns out it could be a solid. And the example I'll show in the webinar here is the solid nanocrystalline dispersion technology for growing nanocrystalline drug from a solid solution. Also, just for completeness, the amorphous drug polymer nanoparticle dispersions are often precipitated from solution in what could be considered a bottom-up approach as well.
And Mike, while you're transitioning to the next slide, I realized that I neglected to address how questions can be answered. So please, if you have a question, submit at any time using the Ask a Question tab on the left of the screen, and we'll try to answer the questions at the end of Mike's presentation. So sorry to interrupt, but thanks.
Great. Thanks, David. So of the three technologies I'll describe, the top-down, bottom-up nanocrystalline drug and the amorphous drug polymer nanoparticles, the top-down crystals are the only one of those, to our knowledge, that has been incorporated into products for oral bioavailability enhancement, though the other two technologies have been used clinically for these applications. And the story is a little bit different if we talk about non-oral products where, for example, drug polymer particles have been commercialized. But this slide shows the 5 oral products that are based on top-down crystals, and 4 of them are based on the nanocrystalline milling approach and 1 on the IDDP homogenization approach. And phenofibrate is represented twice, 1 from milling and 1 from homogenization. And there are a few nice summaries of these products in the literature, and 2 of those articles are cited here, so I encourage you to look at those if you're interested in some details. Since the milling approach is maybe the most widely used attrition process for nanosizing drugs, I will focus on that one in describing the top-down approach. What is shown on the top portion of this slide is a schematic of the physical situation in a typical milling application. Drug crystals, along with some surface stabilizers, are added to a chamber with milling media, and energy is input to fracture the crystals. The concentration and physical properties of the API, the amount and type of stabilizer, and the size and type of grinding media can all be important in this process. The milling media can be ceramic beads such as yttria stabilized zirconia, or cross-linked polystyrene has also been used. Typically, the drug concentration is between a few percent and maybe 20 weight percent in the suspension. To stabilize the crystalline surface, it's common to use both an amphiphilic polymer in combination with a surfactant. And some examples of those stabilizers that have been used frequently are shown on the left part of this slide. Choice of stabilizer for a particular application tends to be fairly empirical at this point. The specifics of the actual milling process depend on the scale and the equipment available. At a small scale, relatively simple grinding chamber is used, and the energy to agitate the mixture is often provided by shaking the entire chamber to achieve attrition. At larger scale, it's more practical to have an agitator inside the chamber. For example, the mixing blade shown in the schematic here. In addition, mills can be operated in either batch or continuous mode. At small scale, typical for early feasibility studies, it's usually batch processing. At larger scale, the process is designed for continuous processing using a recirculation loop to control the mean residence time in the chamber. A typical mean residence time to achieve nano-sized crystals is between 30 minutes and 2 hours. And the graph shown here at the bottom of this slide shows a D10, D50, and D90 particle size in a typical result that's been achieved in our own laboratory. The particle size as a function of time can be modeled, we found, based on a mixing term coupled to the particle breakage constant. So we've done some of that work as well. For early feasibility studies, drug crystals would typically be made at a small scale in a batch mode with tens or hundreds of megs of API using a small chamber that is agitated by shaking the apparatus. A typical approach for a feasibility program that we would use here at Bend would be to use about 5% API in an anti-solvent, which is usually water for the low solubility compounds that we normally use. A formulation screening is carried out in which the API concentration and the type and amount of stabilizer is varied. A number of formulations can be made at the same time for screening. The suspensions are characterized by a range of techniques, including dynamic light scattering, polarized light microscopy, filter potency, and SEM to assess the size and distribution and morphology of the particles. Then the performance is typically tested in an in vitro dissolution test to confirm the degree of dissolution rate enhancement. Usually at an early stage, some initial stability studies are performed to determine whether the crystal ripening or aggregation occurs in suspension over a period of days or weeks. These materials can then be used for animal PK studies to assess the improvement in bioavailability resulting from dissolution rate enhancement. In our experience, it's frequently possible to produce submicron crystal sizes, often in the few hundred few hundred nanometer range using this approach. At the smallest scale, the yield from this process can vary quite a bit and is often not high. But as the process is scaled up, the yield tends to be above 90%. Typical mills and associated scales are shown here in this slide. The Dynamills and also the Nesh mills are commonly used at the larger scales, and they can be operated in batch or continuous mode, as I mentioned earlier. At the early stages of formulation development, the nanocrystalline suspensions are often used directly as suspensions in vitro or in vivo tests, but the suspensions can also be reduced to a dry powder by a variety of methods, including rotoevaporation, fluid bed processes, or spray drying. A powder form can be useful both to stabilize the suspensions for storage and for producing solid dosage forms. I'll talk a little bit more about drying and resuspension of nanoparticle suspensions a little later in the presentation in the context of drug-polymer nanoparticle suspensions, where many of the key considerations are similar to nanocrystalline suspensions. So before moving on to the other two technologies that I wanted to mention, I wanted to return to the developability classification system to talk a little bit about where the different technologies are likely to be most useful. As I mentioned earlier, there are cases where absorption is dissolution rate limited and others where absorption is solubility limited. One way to assess that is to estimate the maximum absorbable dose, which is actually the basis of the quantitative version of the map shown here. So several formulas exist for estimating the maximum absorbable dose, and these have been described in the literature. Dressman uses a so-called solubility-limited absorbable dose. I show here a slightly different formula for the MAD from Curatolo and co-workers involving the product of the drug solubility in the intestinal media, an absorption rate constant, the volume of fluid in the small intestine, and the small intestine transit time. So looking at the right-hand side of the slide, if the MAD is equal to or higher than the actual dose that we want to deliver, then a disso rate enhancement may improve absorption over what is achieved from a typical formulation based on bulk crystalline drug, for example. If, on the other hand, the MAD is much lower than the actual dose, then the dissolution rate enhancement won't help you get the full dose absorbed, and increasing the dissolved drug level is what's probably needed as a path forward. There are varying perspectives in the scientific literature, but I think there's a bit of a consensus these days that the size crystals obtained using top-down attrition processes that I just described, citing the 150 to 400 nanometer range, mainly increase the dissolution rate due to the increase in surface area, as shown in the gray box there, and perhaps by decreasing the diffusion layer thickness, H, shown in the denominator of that equation. But usually not increasing the solubility of the drug. Smaller crystals, which are potentially achievable through a bottom-up approach in amorphous systems, such as the drug-polymer nanoparticles I'll describe, can enhance not only the dissolution rate, but also the activity, or at least the transient solubility of the drug, as suggested in the green box on the right-hand side of the slide here. So I'll move from nanocrystalline suspensions to amorphous drug polymer suspensions. These nanoparticles consist of drug molecularly dispersed in a water-insoluble polymer matrix, usually using a stabilizer such as a small molecule surfactant. High glass transition polymers such as cellulosics or acrylates are normally used to help maintain physical stability in the amorphous state. I'll describe a couple of the commonly used processes, although there can be quite a few variations, and I won't try to cover every possibility. These particles are usually produced in one of two ways, either through an oil-in-water emulsion process shown at the top of the slide, or direct precipitation from mixing solvent and anti-solvent, as shown on the bottom of the slide. In the emulsion process, the drug and polymer are added to an oil phase, which is normally a water-immiscible, high-volatility organic solvent such as ethyl acetate or dichloromethane. Often an ionic or non-ionic surfactant is added to the water phase to stabilize the emulsion droplet. There's typically a pre-emulsion step using a rotor stator or similar mixer, followed by emulsification in a microfluidizer or piston gap homogenizer. After emulsification, the organic solvent is removed by evaporation or through solvent exchange to give a suspension of nanoparticles. For the precipitation approach, the drug and polymer are dissolved in a water-miscible organic solvent and then mixed with an aqueous phase, which is the anti-solvent. Many times there's a surface active agent in the aqueous phase to stabilize the particles here as well. The mixing can be done in various geometries. Shown at the bottom of this slide is a simple T mixer that we have used. And then afterwards, the solvent can be removed again by evaporation or exchange. In choosing between these two processes, there is more flexibility in the solvent in the precipitation process, but higher concentration suspensions are easier to produce using the emulsion process typically. Of course, the suspension from either process can be concentrated by removing water. Since the time required for solidification of the emulsion droplet is on a longer time scale than for the precipitation process, typically minutes or hours even, versus a fraction of a second for the precipitation process, The drug and polymer have a longer time to organize themselves than the emulsion droplet and can produce a different particle architecture with respect to core shell properties, for example, than in the precipitated particles. And we've characterized some of these differences in our own laboratory from these different processes. Like the top-down nanocrystalline drug, these drug polymer nanoparticles can be made at very small batches using just 1 or 2 megs of API in some cases in a milliliter or 2 of suspension media, using a small-scale microfluidizer or a precipitation technique. We show a home-built microfluidizer here on the left side of this slide. And then they can be scaled up to the many kg active scale for either the emulsion or the precipitation process. The nanoparticles made in this way are typically on the order of 100 or 150 nanometers in diameter, as characterized by dynamic light scattering. They tend to be spherical in shape, as shown in the cryo-TEM images shown here. The drug is in the amorphous state, and so the PXRD, not shown, typically shows a lack of diffraction peaks. By modulated DSC, one can typically see a single TG that is intermediate between that of the polymer and that of the drug, suggesting a good molecular dispersion has been made. One advantage of the drug polymer nanoparticles is the higher and adjustable drug activity related to the relative to crystalline solubility. So the physical model that describes pretty well the drug activity is a partitioning of drug between the polymer matrix and the aqueous use environment, as shown in the schematic of the upper left of this slide. The drug will have some solubility in each of the 2 phases, that is the polymer and the aqueous phase. And drug will come out of the polymer phase until the drug activity or concentration relative to solubility is the same in the polymer matrix and in the aqueous phase. So the higher the initial loading in the polymer, the higher the activity in the aqueous phase, as shown in the non-sync dissolution test here in the upper right of this slide. From this plot, you can see that the activity in the aqueous use environment can be either lower or higher than the crystalline solubility, depending on the drug loading and the solubility of the drug in this polymer matrix. Not surprisingly, the release rate of the drug from the particles tends to be rapid due to the very short diffusion distance for the drug to transit out of the particle. The release can be measured in MOVL, and the times tend to be in the sort of seconds. So the amount of drug that comes out of the non-sync in the non-sync environment depends on the activity, but whatever does come out typically comes out very quickly. This slide has a little bit more detail on the drug activity of these systems. As I said, the solubility of the drug in the polymer is important and determines the drug activity versus drug loading. And the solubility of the drug in the polymer can be estimated by calculating the free energy of mixing using solubility parameters, for example. And you can see for 4 model drugs shown here in the upper left-hand plot that the drug solubility in the polymer, which is near the minimum of the free energy curve, can vary quite a bit. The lower the solubility, the higher the activity at a given loading. So the upper right-hand plot in this slide then shows the resulting measured activity as reflected in the dissolved drug levels versus loading for these 4 systems, which can seem to track the solubility of drug in the polymer. So the higher curves result from the lower drug solubility in the polymer. When the loading gets too high, of course, so the red points shown in the upper right plot, then the drug crystallizes, typically during the manufacture of the nanoparticles themselves or soon thereafter. So the physical stability with respect to crystallization can be further assessed by sort of aging dried particles at high temperature and humidity, and you can see on the bottom plot there where the highest loaded compositions, which are in the kinetically unstable region in the upper right plot, are shown to crystallize as evidenced by the drug melt in the age sample of the 25% loaded nanoparticles shown in the red curve on the DSC at the bottom of the page. So the activity and stability is kind of tunable and understandable, I guess, is what we're trying to say here. I mentioned that nanocrystalline as well as drug polymer nanoparticle suspensions can be dried by several methods in order to process them into solid dosage form, for example. One convenient approach for doing so is spray drying. Depending on the formulation, usually a water-soluble matrix material is added to the nanosuspension to aid in later resuspension of the dry powder. And then the suspension is fed into the spray dryer. The resulting spray-dried powder can then be put into a capsule or compressed into a tablet, for example. Making a resuspendable powder of nanoparticulates of either crystalline or amorphous particles often requires an appropriate water-soluble resuspending matrix. Many different resuspending matrix materials will work if we're willing to dilute the particles enough in the matrix, but often we want to minimize the amount of matrix material so that the drug loading in the powder is maximized. What we found is that the small, highly water-soluble molecules, such as simple sugars, often are not the most efficient for suspending the particles because those materials tend to wet and dissolve quickly, leaving behind some stranded aggregated nanoparticles. And this is shown schematically in the first row of the upper right image in this slide. And there's an associated TEM image showing the aggregated nanoparticles resulting from a trehalose matrix, for example. On the other hand, large water-soluble polymers are sometimes prone to forming gels before they can disperse. And so that can not be very efficient for redispersing these nanoparticles either. The example there is shown in the middle row of the resuspension schematic in the right of this slide, where we've used a high molecular weight polymer. And the TEM images show the gelled particles there as well. So what we found it works pretty well is using relatively low molecular weight amphiphilic polymers that are charged, as these tend to be very efficient at dispersing nanoparticles, particularly if the nanoparticles of polymers have a similar charge. For example, a negative charge from acidic groups. Examples of matrix materials that we've used are CMEC and HPMCAS, as well as some biopolymers. Shown in the bottom row of the schematic at the right and in the bottom TEM image is a resuspension using this type of polymer, which works quite well at relatively low levels. The plot at the bottom right of this slide shows good resuspension of nanoparticles in a simulated gastric to simulated intestinal fluid transfer dissolution test, in which the nanoparticle resuspension is assessed using filtration through a 0.2 micron membrane. And this test shows that we get very rapid redispersal of nanoparticles from a spray-dried powder using either CMEC or HPMCAS respectively. So resuspension is feasible using a minimal amount of matrix material, say 1-to-1 suspension matrix to nanoparticles, or slightly less in these applications. The last technology that I want to touch on is a bottom-up approach for forming nanocrystalline drug. And I'll refer to this as solid nanocrystalline dispersions. The idea in this particular approach is to make a spray-dried dispersion of drug in a high TG polymer and then age the dispersion at high temperature and humidity, which imparts mobility to the SDD matrix, allowing the drug to nucleate into small crystalline domains. As the drug comes out of the dispersion, the glass transition of the increasingly polymer-rich matrix then rises, shutting off the mobility of the drug and preventing ripening of the crystals. This results in polymeric particles of tens of microns in size having dispersed within them drug nanocrystalline domains of maybe tens of nanometers in size. And the PXRD of these materials, an example of which is shown on the bottom left of this slide, typically show very broad diffraction peaks indicative of the small crystalline domains. They're achievable using this approach. This slide shows in a little bit more detail the crystallization process, which is kind of key to the technology. In this example, we have a drug polymer dispersion with a high TG polymer and a low TG drug. We place the SDD in a temperature in RH such that it is above the TG of the dispersion, resulting in a lot of drug mobility. And then the drug begins to migrate and crystallize into small domains. And as it does so, the TG of the increasingly drug-poor matrix continues to rise. And so all the drug is crystallized and the TG is that of the pure polymer. And the annealing conditions are chosen so that when we've reached this point of complete crystallization, then the mobility of the polymer matrix is low enough that there's little or no ripening of the crystals that we've formed. So one advantage of the solid nanocrystalline dispersion approach is the ability to control the ripening by shutting off the drug mobility through the rising TG of the polymer-based matrix. In precipitation from liquid solutions, on the other hand, preventing growth or ripening of the nucleated crystals is often a significant challenge, and the surfaces have to be stabilized carefully with surface stabilizing agents. It's very advantageous, of course, to have very small crystals if they can be stabilized, because if they're small enough, then these crystals can have higher activity or transient solubility than the bulk crystalline drug. So the process we show here is a two-step process, that is spray drying and then annealing. I want to note that we're also working on making similar materials, that is small crystals in a solid matrix, using a single unit operation. And that approach is something that we might be able to talk about sometime in the near future. I wanted to talk a little bit about solubility versus crystalline size, because it's discussed a fair amount in the literature and in the industry. The expected solubility of small crystals is often expressed in terms of the Ostwald-Freundlich equation, showing the predicted log of the ratio of nanocrystalline solubility to bulk crystalline solubility in the top of this slide, which is inversely proportional to the crystal size. So, and the important surface tension term, gamma, if in the numerator, can be calculated using the expression on the right-hand side of the slide, and is derived from the Gibbs-Thomson equation, in which the melting point difference between nanocrystalline and bulk crystalline materials and the heat of fusion can be measured using differential scanning calorimetry. And then the radius of the crystals can be estimated from the Scherer equation from the PXRD diffraction pattern. And some of these examples of some drugs, 5 drugs, are shown in the table in the middle of the slide. The plot in the bottom of the slide then shows the surface tension values for several solid nanocrystalline dispersions that have been obtained using this approach. The surface tension values that we plot here are consistent with others reported in the literature, and a second curve is shown on the plot, which is the surface tension calculated using a semi-empirical equation for drug solubility, which also makes use of the melting point depression of the nanocrystalline domains relative to bulk crystals. So that's also plotted there in the figure, and is in good agreement with the calculated data based on DSC and PXRD, and also from what we see in the literature. So once we have those values, we can make plots, and so based on those surface tensions, we've estimated the solubility versus crystal size for these specific compounds made through this process. And the results here, you can see, is that to significantly increase the activity, say to 2 to 3 times the crystalline solubility, the crystal size has to be down below 100 nanometers or so, which, as I said before, is not easily achievable using the top-down approaches for making a nanocrystalline drug. So also note that for a given crystal size, a lower surface tension gives a lower solubility. And then in the top-down approaches, we go to great lengths sometimes to lower the surface tension by adding surface stabilizers, where in the solid nanocrystalline dispersion approach, the crystals are stabilized not so much through optimizing surface interactions, but by lowering mobility in the polymer matrix to shut down crystal growth. So in principle, at least, higher activity crystals of a given size can be made using this approach versus another approach where we really have to stabilize the surface. Okay, so now in the time remaining, I just wanted to go quickly through a case study for each of the 3 technologies that we've presented today, and I'll start with the top-down milled nanocrystal example from the literature. This is a nice study in which the authors were looking for increased bioavailability through increased dissolution rate by attrition milling of crystals. And they showed data for 3 different crystal sizes made by hammer milling, jet milling, and then nanocrystal milling, respectively. And you can see the size distributions here in the upper left of the slide, and then the dissolution rate differences in the upper right, which track nicely with the sizes. And then they showed progressively increasing oral bioavailability as the crystal size gets smaller. So if you go back to our maximum absorbable dose calculation we were talking about earlier, the increase in absorption in going from the hammer milled to the jet milled material makes a lot of sense based on overcoming dissolution rate limitations in absorption. However, it appears in this case the significant increase in bioavailability for the nanocrystalline suspension might be due to another factor besides dissolution rate enhancement, as the MAD calculation suggests a maximum absorbable dose of maybe only 10 to 20% of the 100 mg that was administered to the dogs here. So there may have been something else going on here, maybe a form change or solubilization from surfactants or something that might have increased this transit solubility of the drug compound in that particular nanomilled formulation. The second case study that I'll go through is of an amorphous drug-polymer nanoparticle for Celecoxib in ethyl cellulose made by the emulsion process that I described earlier. So the formulation driver here was rapid absorption of this drug for pain management. And the nanoparticles that were made consisted of as I said, Celecoxib and ethyl cellulose, and were made by an emulsion approach using either dichloromethane or ethyl acetate into water. And these were about 100 to 150 nanometers in diameter as measured by dynamic light scattering, and were spherical as mentioned before, as shown by the TEM. They had a TG suggesting a molecular dispersion, and as you can see in the bottom right-hand side of this slide, were resuspendable after spray drying with HPMCAS, CMEC, or casein in this example. These particles were then tested in dogs and in humans. In dogs, the original suspension and a resuspension from a spray-dried powder had similar performance, both having much higher exposure than the commercial capsule. And this data is shown on the left-hand plot on this slide. In humans, the resuspended powder likewise had higher exposure than the commercial capsule, and also more rapid absorption than a spray-dried dispersion of Celecoxib with HPMCAS, and also more rapid than the commercial capsule as well. And I think David mentioned in an earlier webinar that you know, the effective concentration of this drug is a few hundred nanograms per mil, and for a pain indication. And so in this case, getting rapid absorption of drug from the nanoparticles quite possibly leads to a more rapid onset of pain relief, and so it could be very advantageous therapeutically for that type of an application. The last case study is one for the solid nanocrystalline dispersions of Ziprasidone in HPMCAS. This solid nanocrystalline dispersion was investigated to increase bioavailability, particularly in the fasted state, and it was used because it's very difficult to stabilize the amorphous form of the drug due to the strong tendency of Ziprasidone to crystallize. And you can see it has a very high melting point of 321 degrees C. The broad PXRD peaks here are indicative of small crystalline domains, and there's a recrystallization peak in the DSC you see on this slide in the lower left for the STD, but not for the solid nanocrystalline dispersion once the drug is crystallized out. And this suggests that the vast majority of the drug is crystalline in that solid nanocrystalline dispersion. The lower right-hand part of the slide shows a biphasic sink dissolution test, which suggests higher dissolved drug levels in the test for the solid nanocrystalline dispersion versus the bulk crystals, as evidenced by the sharper slope for those formulations in both the simulated fed or simulated fasted media. In dogs, the solid nanocrystalline dispersion of Ziprasidone mitigated a twofold fed-fasted effect that was seen with the commercial capsule. And the solid nanocrystalline dispersions, of course, can be used in a variety of dosage forms. It can be granulated, put into beads, or put into IR or osmotic tablets. here is an FEM image showing a granulation that was done, and it was put into a tablet and dosed in a clinical study that, like the dog study, showed a reduced food effect for this drug. So just to kind of wrap up and summarize then, I think we've shown that there are a number of nanotechnologies that are useful for increasing bioavailability of poorly soluble drug compounds. Nanocrystalline drug particles and drug-polymer dispersions are two technologies that have been used successfully to demonstrate clinically and commercially this bioavailability enhancement. And a fundamental understanding of the physical situation, for example, whether a situation calls for dissolution rate enhancement or solubility enhancement to improve absorption, allows rational selection of formulation technology and guides development of new technologies as well. So with that, I'd like to stop and thank the audience for their participation today, and we'd be happy to answer any questions.
That concludes our presentation, and thank you, Mike. I see that we have a number of questions here, so let me get started with the first one. Mike, here's a question. Are all three of the technologies described commercializable? And what barriers exist for scale-up and commercialization?
Yeah, thanks, David. That's a good question. So the three technologies we went into some detail on, the top-down nanocrystals, of course, have commercial precedence, and there are a handful of products there, as we've mentioned. I think some evolution has occurred in that space with respect to the key players there, but there are partners for scale-up and manufacture of these, so I think that there's a ready commercial path there. On the drug-polymer nanoparticles, there's some precedence in the injectable area for commercializing those types of structures, and I think that all the equipment and processes are available for commercial scale manufacture. It's still a relatively new and developing area, so I think that with respect to the key partners there's probably a little bit more of a challenge in identifying and establishing those. The solid nanocrystalline dispersions, of course, consist of two unit ops, the spray drying and then the annealing. And of course, spray drying is commercially available. And in fact, we here at Ben DFS are building additional capacity for commercial spray drying. And then the annealing at the commercial scale is available. I think the main challenge on that one is the process optimization and robustness to choose the annealing conditions. And so as we sort of alluded to, combining those two unit ops into one may be advantageous with respect to smoothing that manufacturability going forward.
Okay, great. Thanks, Mike. The next question, interesting. Do drug polymer nanoparticles or nanocrystals get directly absorbed?
Yeah, I think maybe this question comes from maybe some reports in the literature where people are actually trying to decorate nanoparticles with ligands to get them orally absorbed for specialized applications, typically not for bioavailability enhancement. So I think that there are some reports here and there of that for things where people go to lengths to decorate those with ligands, maybe to get them absorbed through the M cells or other specialized cells. But I think the particles that we've talked about here, the nanocrystals particularly, but then also the types of drug polymer nanoparticles, we don't believe would be appreciably absorbed into the systemic circulation.
Great. Follow-on question here. Is lyophilization good, bad, as compared to spray drying to get powders of nanosuspension nanoparticles?
Yeah, of course, we didn't really talk much about lyophilization, but both lyophilization and spray drying, which we did mention, are great methods for removing water or other solvents for reducing these types of particles to powder. Lyophilization has been used quite a bit. Spray drying is a continuous process and maybe a little bit easier to scale than lyophilization, but they both have kind of their pluses and minuses. So certainly lyophilization is used quite a bit as well.
Here's a question, again, around the process. Does the wet milling process result in semi-crystalline materials?
Yeah, I think that's a very good question, because I think we alluded to in talking about the case study of the nanomilled material, where there may have been something else going on besides just size reduction. Oftentimes we do worry about creating amorphous material by milling things with all the heat and energy that's put into that. Or changing the crystalline form as well. So either making amorphous material or changing the crystalline form is often a concern when milling, and so that's typically something that you might want to check so that you know what you end up with at the end. So good question.
Looks like I can handle this next one. Are you planning on having a similar webinar on your modified release technologies? If so, is it already scheduled? The answer quickly is yes. And while it hasn't been specifically scheduled, we're expecting to have that sometime this summer. Back to the topic, can you give some examples of injectable nanoparticles, and will your manufacturing steps apply?
Yeah, so there are a number of different types of injectable particles that can be produced. Obviously, one of the big differences there is the excipients that are usable, and also, of course, the sterile processing. But some of the processes and the architectures we've described can be used for injectable applications as well, but there are some additional considerations, obviously. To date, we here have not gotten into the injectable area, but there are people who do that.
Okay, here's an interesting question. Have you compared agglomeration rates between top-down and bottom-up nanoparticle processes?
Yeah, that's a good question. I think that it depends on what the particular processes are. I think that that's a pretty big, there can be a big difference depending on the process as well as the size of the crystals. It's a pretty significant concern for the top-down approach. For the bottom-up approach that we described, which is growing crystals within a matrix, it's not as big of a concern there if you can effectively separate the domains where the nanocrystals are growing. So I think that we've taken approaches to try to minimize that by keeping them separate in the solid matrix. But that is obviously one of the largest concerns with respect to nanocrystals is the aggregation of those. And so typically, matrix materials are used to keep them separate, whether they're made by top-down or bottom-up approaches.
Okay, Mike, and I think we have time for one more question, and there are still many questions that we haven't had a chance to address, and we will follow up with those offline over email. So thank you again for your interest in your questions. Last question. With your drug polymer nanoparticles, when you test the dissolution at increasing drug loading, do you perform 80K ultracentrifugation to get the absolute solubility, or simple 200 nanometer to 450 nanometer filtration to also include nanoparticles?
Yeah, so that's a pretty specific question on kind of the technique, but it points to a significant issue, which is trying to separate, I'll say, freely dissolved drug from small particles that may contain drug. And so we use a variety of methods to try to do that. One is filtration through membranes. One is, of various sizes, so from 1 micron to 0.45, 0.2 micron filters, as well as the smaller membranes that were mentioned. For the data we showed, those were actually went through the say 100,000 molecular weight cutoff filter membranes that are associated with centrifugation. So there are a number of methods of separating kind of freely dissolved drug from drug and particles, and so care needs to be taken to do that, to verify that one knows what's being measured. So good question.
Okay, thanks, Mike. And as I said, those are all the questions we have time for today. And again, questions that we weren't able to get to, we'll follow up offline over email. So we thank you. We do plan to host additional webinars in our Bioavailability-Enhancement series and hope that everyone will continue to participate. The next webinar in the series will be held on March 12th, and we'll cover the hot melt extrusion formulation and process, aspects around performance, stability, and manufacturing. Please check the Capsugel and Ben Research websites for details and to register. On behalf of Capsugel DFS and Mike, I'd like to thank you for joining us today. And if you'd please fill out the survey that will pop up on your screens as soon as the webinar is over, that will help us in understanding what topics are of interest and to be able to have pertinent webinars in the future. Thanks again for your time and attendance today.